Good morning or good afternoon, everyone. Can you hear me? Hi. Thanks for joining online or here in the amphitheater. So it's our pleasure for this fourth session of this infectious diseases seminar to welcome Jean-Michel MOLINA, with whom we've had the privilege of interacting and collaborating with, but we are not the only ones in this room or in this center. Jean-Michel, as you can see from his title slide, has been around for a while doing HIV research.
Too long, maybe?
Too long? No, definitely not. And has been very active, I can tell you that, in collaborations. And so it's our privilege to welcome him. Jean-Michel MOLINA is a professor of infectious disease. He's a clinician and a researcher. He's the head of the infectious disease department of Saint-Louis and Louis-Lariboisière Hospital, APHP, and he's well known in the field, and as you see, and this is why we invited him he has a unique perspective on what has been happening in HIV research, because he's been around for 35 years and changing a lot the landscapes, and he was also instrumental to a recent important change in public health, which was recommending PrEP, which I think was a bold move from our Minister of Health at the time, and so all of this we were very interested to have Jean-Michel's perspective on the spectrum of researches he has been conducting. So Jean-Michel, thanks a lot for accepting our invitation, and you have the floor for this presentation. Thanks.
Thank you. Thank you very much, Olivier. It's a pleasure to be here with you today, and it's an honor to address your group, your team, and a number of people I know very well over the years, and those online joining for today. You know, it's going to be a special presentation today, because I'm not going to talk about a specific topic. And as you asked me, Olivier, to do, I will try to walk you through my career in terms of research in HIV. And that's why I entitle my talk 35 Years of HIV Research, Review and Prospect. And I'll spend a lot of time talking about review, and maybe at the end, and after my talk, we could address the prospects. So I'm a clinician. I lead the Department of Infectious Disease in Paris, and I'm also linked to an INSERM unit doing basic research in virology. So let's go for about 40 minutes of presentation, and then I'd be happy to have a discussion with you all. So when it started, I thought I would show you this first publication in 1958, because that's the year when I was born. And it was interesting for me to see this publication from another Molina, and you could see some resemblance, because that's my father, actually, who was a professor of pneumology in the University of Alger. And he was, in his career, he has worked a lot on TB. And that's interesting because, as you will see, I also do some research on TB. And because of the war, my father then went to the University of Clermont-Ferrand, and that's where I did my medical studies, at the University of Clermont-Ferrand. I was very happy here, very nice University of Medicine. Then I thought, Clermont-Ferrand is a nice city, but if you want to do research, you better go to another place, at least back in the 1970s. And then I decided to try to go to Paris for my residency. And at that time, I did my residency in Paris then. I had a choice, Clermont-Ferrand and Paris, and then I decided, well, let's go to Paris. I had a great time initially, and I did my residency in Paris from 1981 to 1987. And at that time also, I did some training at the Pasteur Institute on virology, microbiology, and epidemiology of infectious diseases. And you have my first publication in English during my last residency stage in Paris on cryptococcal infection in a patient with AIDS, how to make the diagnosis. That was in 1988, my first publication in English. Then I went to the US for my postdoctoral fellowship. I started my PhD in Paris, and then I completed the PhD and the postdoc at the Deaconess Hospital, linked to the Harvard Medical School, for 2.5 years. And I met my mentor at that time. We're still active, Professor Jerry Groupman. And he's an hematologist, and he was one of the pioneers of HIV research in the US, because initially a lot of hematologists actually were working on HIV. And I learned about hematopoietic growth factors, the interaction between HIV and the bone marrow, how to look at the infection of progenitor cells, also macrophages and the interaction between HIV and the cytokines. And thanks to him, I had my first paper in the New England Journal of Medicine in 1990, so more than 30 years ago, on the biology of growth factors. And we did a couple of publications during my time on looking at the interaction between HIV and the macrophages, the cytokines, and also looking at how to grow progenitor cells from the bone marrow. That's an interesting assay to set up. And then we looked at whether or not the bone marrow cells, the progenitor cells, would be infected with HIV, which is important when you think about cure strategy for HIV, because if the progenitor cells are infected, that's an issue. And we couldn't find any infection of these progenitor cells at that time. Then it was time to go back to Paris in the Department of Infectious Disease at the Saint Louis Hospital, and my mentor here was Professor Modai, who has passed away, unfortunately, due to COVID in his department a couple of years ago. And at that time, during my what's called Clinica, I was very busy with the clinical management of patients with AIDS, and we had a lot of young people dying of AIDS at that time. 1996 was before we had, as you know, very active antiretroviral therapy available. Many people had opportunistic infection, and we have very high morbidity and mortality rates. So at that time, I was interested in looking at opportunistic infection in general, and in particular on this one, microsporidiosis, which is a very broadly, very ubiquitous protozoal infection that usually gives you intestinal infection and diarrhea. And I was really impressed by one of the patients I had at that time, who happened to be the architect of the Hôpital Saint-Louis. And these women actually was dying of intestinal microsporidiosis because of chronic diarrhea, wasting, weight loss, etc. And I thought this was a very, at that time, important infection to try to better understand. And, we first published the clinical features of this infection to try to understand what was the problem. And so with a group in Hôpital Saint-Louis, our colleague from the gastroenterology, we identified the parasite for the first time, and then tried to define the clinical spectrum of the disease. And then I set up with Professor Derouin, who also has passed away now more than 10 years ago. We set up a research unit within the university to address this topic of intestinal macrosporidiosis, because very few teams at that time were working on this infection. And so we improved the diagnosis of infection. Initially, you had to perform duodenal biopsies, and then we set up PCR assays to identify this parasite, looking at PCR first in duodenal biopsy, then in stool samples. And we thought that this parasite was not unique. There was many different subtypes of this parasite with different clinical spectrums. And then we said, well, we have to, I'm a clinician, as you know, and we were thinking about, now we need to find a cure, a treatment, because there was no treatment for this parasite. And we tested different drugs with different clinical studies. With other types of microsporidia, we could find very effective response with albendazole in randomized placebo-controlled studies. So that was one thing. So with two different parasites, we could establish the effectiveness of albendazole. But for the most frequent enterocytosome BNUZ, we couldn't find any treatment. So we had to try to test a number of different drugs, and we did that in a phase 2 study where we tested, 10 patients with one drug. Then, if it didn't work, then we went to another drug until we could find an effective drug. And the effective drug was fumagillin, which is a drug that was found doing literature review. I did an extensive literature review to try to find drugs that could be appropriate to treat this disease. And I went back to the 1960s, where this drug was used for the treatment of enterocytosome or other type of microsporidiosis, which were infected a number of animals, because it's very common infection in animals. And I can't find the word in English, but this was a parasite which causes a lot of problems in agriculture. And at that time, they were using fumagillin to treat these animals, and it worked very well. In human, it has been also used for the treatment of intestinal amoebiasis at that time, before we had the drug we use today. And I thought this is a drug that we might be interested to test in our patients. And I'm telling you the story because that's interesting. Then I tried to see whether the drug was still being used for treatment in veterinary disease, and in fact it was, to treat honeybees, because if honeybees are infected with microsporidia, they do not produce honey anymore, and that could be an issue. And then I contacted the company, Sanofi, who was still making the drug, to see whether we could make a partnership to try to see whether we could have a drug that could be used for humans. And when a drug is available and appropriate for animals, for bees, it's not exactly immediate to use the same drug for humans. So we had to work with Sanofi and the French Drug Regulatory Agency to make sure we had all the requirement to be able to test this drug in humans. And when we did our screening trial, this was the only drug which was magic. Because with all the other drugs, you couldn't eradicate the parasite from the stools. And with that drug, the first patient cleared the infection within 2 days, and he resumed gain or he gained weight, 10 kilos in about a month. So that was kind of spectacular. I forgot to mention that in the lab, in vitro, we replicate the cycle of this parasite, and we tested the drug in vitro as well. In vitro, it worked very well. That's why we were encouraged to test it in humans. Then we try a dose-ranging study to try to define the best dose to use. And when the best dose was found, then we did the placebo-controlled study that we published in the New England Journal in, I think, 2002, I think, in this range. And the drug, because at that time, because of the availability of highly active antiretroviral therapy, we had fewer patients with very advanced immunosuppression. So we had fewer patients with HIV, and so we included also all the immunocompromised patients, in particular bone marrow transplant patients or patients with renal transplant, in the study. And the study was a frank success. You had 100% success in those receiving fumagillin and 0% success in those receiving placebo. And when you switch the placebo to fumagillin, then all cleared the infection. So there was a drug with 100% effectiveness. And the drug is still being used now, not anymore in patients with HIV because the infection has disappeared today, but is being used for the treatment of renal transplant recipients. And so we did, and even recently, we published in 3 years ago, a review of all the patients in France who received the drug for treatment of intestinal microsporidiosis in patients who have received a solid organ transplant. And for this drug, I had a patent, and this patent, gave me some revenue, which was interesting to note because the drug was very expensive. But this is another story. So we could discuss that later. At the same time, I grew up in the department and became a professor of infectious disease. And there was, at about the same time that we had highly effective antiretroviral therapy available. And then I started to be more interested in trying to improve what we had in terms of treatment for HIV infection. So I became the head of the department in 2001. And that's important. I was really involved in the network ENRS, the National AIDS Research for AIDS and hepatitis. And I led within this agency the clinical trial group from 2005 to 2017. And it was very interesting to have this interaction with other colleagues in other fields of medicine, our colleagues from microbiology, from pharmacology, from social sciences, and also with the community of patients. So a very useful and interaction. And then I became involved in clinical trials of antiretrovirals. And my first trial was done actually with Geneviève Chen here, whom you know very well. And we started this collaboration with Bordeaux, so you can see that it's a long-lasting collaboration with one of the trials that we started with Geneviève and her team, with Valérie Journeau, with whom we are working again 24 years later. And we started the first trial, the ALBI trial, just to mention that that was the first trial we did at that time. And then we tried another combination. And I mentioned this combination because that was the first time we used a combination of 3 drugs for the treatment of HIV infection in a single dose, or in a single drug intake, so once a day. Because at that time, and we were, I can't find in the year 2000, we used a combination of 3 drugs with a huge number of pills, 2 to 3 times, even 4 times, why not 5 times a day. And at that time, I had a collaboration with a friend of mine who was working in the US in a small drug company called Triangle, which has been bought by a bigger one, Gilead, today, to test emtricitabine. And emtricitabine is a drug that we tested for the first time. And because of this collaboration, it was okay to test this drug in a French trial, which we called Montana, because it's mono combination once a day. And at that time, I had a lot of discussion with colleagues from the pharmacology field, which were telling me, well, this is not going to work. Once a day is not going to work. You need to give the drugs at least 3 times a day. I say, well, let's try it, because there was no reason why it should be that way. And I mentioned this study because that was, again, the first study done with a triple combination of antiretroviral therapy given once a day. And this study was a very good, provided very nice results with very high rate of success. And because of that, there was a small pilot study. Because of that, we developed additional trials with a much larger sample size, still with the group of Bordeaux here, called the Alizé trial, where we did a non-inferiority study, and we looked at many different secondary endpoints, which I'm not going to mention here today. Another study I'd like to mention here is other trials are conducted with other colleagues from Toulouse on intermittent therapy, because at that time we were thinking that maybe intermittent therapy for HIV could be a solution to some of the adverse events of the drugs, but we know that it's not useful at all. And this was, that study here, I don't know if you can see my arrow, was a study we did with my colleague Yves Lévy on trying to see whether we could modulate the immune system by using interleukin in people with HIV, trying to avoid the use or defer the use of drugs. And we'll come back to that at the end of my talk. So many trials with the NRS, I mentioned those in which I was involved. In particular, the TRIO trial, that was a trial that I designed, but because I was the head of the clinical trial group at the NRS, I could not obviously lead all the trial. But because of my relationship with the companies, I told them this is a study, a combination we should do in France. And they agreed to work together with the NRS and to be able to start this trial. And I mentioned the role of the company, of the industry, because during that time I've been interacting a lot with a number of drug companies which provide me with nice collaboration and also a lot of knowledge, because the company is very rigorous. When they do a trial, they have to meet very clear guidelines if they want to register their drugs. And so there was a great experience to work with companies and also with other colleagues, other experts from different countries. And you can see here that I've been the principal investigators of a number of trials, looking at DDI or, in particular at that time, atazanavir, ritonavir, which was one of the first PI that was given once a day. Then rilpivirine, which is a drug that is still being given today, as you may know, as a long-acting drugs. Also tenofovir alafenamide, or trying to assess lower dose of ritonavir. And more recently, I'm still interacting with industry to address the effectiveness of integrase inhibitors, doravirine, a drug that is a new INSTI. And because of this relationship, there was an opportunity for us when we want to design trials with the NRS to have the relationship within the industry to have drugs available and also, as I said, knowledge. And so the last publication I had was this publication in the New England Journal last year on a new drug, a capsid inhibitor, lenacapavir, in a trial of multi-drug resistant HIV-1 infection. And I was the senior author on this international study, and this was because of the trust of the company in the work we've done over the years. I also worked with another network, which is an important network, and as you can see, having multiple collaboration is really critical. Again, because that enrich you in terms of knowledge and possibility of doing research. So I've been involved in this important study that was called the START trial, which was conducted at the same time as the Temprano study that you know very well here. And because I was involved in this trial, the START trial, which was a large international study to look at the best time to start ART in people who were asymptomatic with HIV infection, I discussed with the head of the NRS at that time, and I said, well, we've got to do a similar study in France as well. And I pushed him to, approve the Temprano study to make sure that the Temprano study would not be deferred too much. And so this was an important study to change the guidelines, the START trial. And then I could, in this trial, publish the a paper in the Lancet about, who are the people who might benefit the most for early initiation of antiretroviral therapy. And, I learned a lot working with this network, although we have conference call usually at 10 p.m. Once a month, so that's not so easy, but that's important to keep this collaboration. At the same time, during these years at ENRS, I started to set up a collaboration with Brazil. director of the NRS at that time, asked me whether or not I would be interested to set up a collaboration with Brazil. I said, yes, I'm interested. In fact, my sister-in-law is Brazilian, and I said, well, that's a great opportunity. My neighbors were Brazilian as well, and so I said, well, let's go there. And we started a very nice collaboration, now it was like more than 10 years ago, with my colleague Beatriz Greenstein in the Fiocruz Foundation in Rio. So I had the opportunity to go there multiple times. And it happens that one of my closest colleague in my team, Nathalie de Castro, you have this, her picture here, is also from Portuguese ancestors, and so she speaks fluently Portuguese and English. And so that was also interesting to have someone with me to bring in this collaboration. And then we address, there was also with the group of Geneviève Chen, the issue of treatment of HIV in people co-infected with TB, first study, and then more recently a second study, this time with Olivier and his group, the Reflect TB True trial, a much larger study to look at the use of raltegravir for the treatment of this co-infection. And during this clinical trial, usually you use the data or the biobank to address other topics like the PK of the drugs or in particular. And then I said, well, using antiretroviral drugs is nice, but we have now very potent and very good combination. We may not need to do any more research here. It's not that interesting as it was in the past. And I thought then the use of these same drugs for prevention could be probably interesting because we don't have a vaccine to prevent HIV acquisition. And what could we do to prevent infection? And so we thought, well, maybe drugs could be used. We had the example of the prevention of transmission from women to their babies. And we discussed with my colleague Jean-Pierre Aboulker a first submission to ENRS in 2008. And the reason why I became interested in this topic was because I was asked by my colleagues in Thailand to give a talk. It was Marc Lallemand who invited me over to go to Bangkok to give a talk on, what's going to be the future of HIV research. And I thought, well, looking at the literature at that time, prevention with drugs would be the best way to go. And because of the experience we accumulated in the use of antiretrovirals for treatment, we said we are well positioned to test this drug for prevention. But we are not the only one. And we tried to find the best time to start our trial. And we started only the study in 2012 because of we wanted to wait for the results of the first trial done by my colleague in the US. So we present the result of this trial in March of 2015. And very soon after that, the authorities in France, in Europe, and WHO approved PrEP for the treatment, for the prevention of HIV infection. Indeed, the first study was done by my colleague Bob Grant, published in 2010, and we were waiting for the results of the study before we started our trial to make sure that our trial could be done. In that study, they use a daily PrEP with one pill of TDF-FTC or a placebo in MSM at high risk of HIV infection. And what they found is after 1 year follow-up, there was a 44% reduction of HIV incidence. That was interesting, but maybe not high enough. That was actually below their expectation. And they found out that the reason why maybe it was not so good was because only 50% of the people had enough of it detected in the plasma. And so we said, maybe if we use another design, not giving PrEP every day, but only at the time of sexual exposure, we could have a better adherence and therefore a better effectiveness. So, but we couldn't do that until we knew that the results from the US trial were okay, but not perfect, and that there was room for improvement. And so we did also a randomized placebo-controlled study in the same population of high-risk MSM in France and Canada. to see whether TDF-FTC given before and after sex versus placebo would prevent HIV acquisition. So that was the scheme of how to use it, with a loading dose of 2 pills. Not to mention that my colleague from the PK expert, I'm not talking about the French PK expert, but the US PK expert told me this is not going to work. No way that PrEP regimen given just before and after sex would prevent HIV acquisition. Well, but in fact, that was an adventure, I would say, this trial. I had to work with the community a lot, so I had the experience working with the community for treatment, and this time that was with the community for prevention, because for prevention, people don't come to the hospital, so you have to find them in nightclubs. As you can see, I'm in the back trying to explain in gay clubs why we're willing to do a trial for prevention, and why they should be enrolled in a placebo-controlled study that was not granted. And these are the participants. You see the picture in the middle. At the end of the study, we celebrate with the participant the result of the trial, which was made possible also by the involvement of the community leaders. You have ACT UP Paris and AIDS here. And indeed, the results were quite striking. When you look at the incidence in red in the placebo arm, it was as high as nearly 7% per year, 9% in Paris. And when you look at the blue line with TDF-FTC, you can see 0 infection during the first 16 months, so almost 100% effectiveness. And only 2 individuals at the end became infected because they stopped PrEP. So the effectiveness was not anymore 44%. It was 100% if you look at people taking the drugs, and 86% in the intention to treat analysis. So the results were published in New England for the World AIDS Day in December of 2015, a few months after the presentation of the results at CROI. And we also published a couple of months later the follow-up results, because in the follow-up study, all participants could have access to PrEP, which was not available at that time in France. And the effectiveness, that's why I mentioned this paper, because we had an effectiveness in a clinical trial of 97% of reducing the incidence of HIV. So that was very high. We had an editorial by Anthony Fauci at that time in the New England saying that we had the tools to end the HIV epidemic. We have to follow the results from the science. We are very happy about that. Unfortunately, at the same time, it was in November of 2015 in Paris, we had a terrorist attack in Paris, very close to our hospital. It was just the restaurants where people used to go for dinner to have a break. So actually the people wondered, and I had a couple of my colleagues from my team in the bars where the terrorist attack occurred, and they had to use their bell to to try to save the lives of these people at that time. And because of that, I mentioned that because, well, we could not celebrate as we wanted the result of the study because of this terrorist attack. And at that time, the editor of the New England Journal of Medicine called me, because we were in the time of publishing our results, and he said, well, I know what's going on in Paris. Is there a way you could help me to do a paper on how you are handling the terrorist attack in the hospitals in Paris? And that's why we had this paper also from my colleagues in the intensive care unit at the emergency room about that. Well, anyway, the results were published in the WHO 2015 guidelines. The Ministry of Health approved PrEP as early as January of 2016. And then we, because of the large number of data, we could publish a number of secondary papers on the cost of these prophylaxis, the issue of drug resistance, the issue of toxicity, and we made sure that as compared to the daily use of PrEP, the effectiveness of Fundament was as good, and we recently conducted a large cohort study in the Paris region, trying to see whether when you implement PrEP, you could have an impact on the new infection at the population level. So we did this large trial which showed that whether you use daily PrEP or on-demand PrEP, you had the same effectiveness in terms of HIV incidence. And we, within this trial, looked also at rectal tissue biopsies as a mean to try to predict the effectiveness of PrEP regimen, which is a difficult assay to set up. And for that assay, we collaborated with our colleague at the Microbicide Treatment Network in the US to help us to set up the assay in Paris. So this regimen of the called 211 or on-demand PrEP has been endorsed also by WHO and by other guidelines. And today, as you may know, the last report from EPFAR showed that in November of 2023, more than 95,000 people have started PrEP in France. And what's nice is that from the most recent data from Santé Publique France, you can see that in red, among men who have sex with men, those who are using PrEP, because 97% of people using PrEP in France today are men who have sex with men, you can see a very nice decline of new infections, new diagnoses, I have to say, 32%, whereas in the other groups, you see almost no change. So we have the evidence that not only this PrEP regimen works at the individual level, but it also works at a population level when you implement and you target the right people. So we are trying now to improve this regimen, and we're working with Geoffroy, one of my young assistant, who is now doing a postdoc in Chicago, working on implementation science. And we have this trial that we will start very soon, we hope, in collaboration with Thailand, because we have kept the links with Thailand, which I mentioned earlier in my talk, trying to assess a simpler regimen for on-demand PrEP, just one pill before and after. And again, with TAF-FTC. TAF-FTC is not approved in France. And again, it is because of the collaboration we have with Gilead that we were able to get from Gilead the drugs to do the trial. Within the Prevenir trial, which I mentioned, we are also going to start a study using cabotegravir, a long-acting drug for PrEP, which is not available yet in France, but the drug has been approved by the FDA in the US. But in France, not yet, because probably of cost issues. And we want to know whether in an open-label study, this drug really provides benefit to the patients and whether it could improve adherence to PrEP, because we know it's a critical issue for effectiveness. And finally, we will start also in France and the UK, probably in August, a study with another drug for PrEP, lenacapavir, given only once every 6 months, so with 2 injections per year, you may prevent HIV acquisition. So it's almost like a vaccine, and probably better than a vaccine to prevent HIV acquisition. Just a word about COVID, because during the COVID epidemic, all these studies were ongoing. And I was very busy as the head of the Department of Infectious Disease at Saint-Louis to take care of the patients. And at the same time, I was also became the head of the Louis-Lariboisière hospital. So I had 2 clinics to take care of. So very busy to take care of the patients, a lot of death again. So I was not involved in a research network on emergent infectious diseases or on COVID-19, because I was seen as probably only an HIV expert and not an expert in COVID. I think nobody was an expert, but I was not therefore involved in the networks. But we did our own research. And just to mention that, in March of 2020, we published a small study we did in our unit to say that hydroxychloroquine didn't work. And the Quotidien du Médecin, mentioned that, because you only heard at that time what Didier Raoult and his group were doing in Marseille. And, these journalists said, there are results from the team at the Hôpital Saint-Louis where they think hydroxychloroquine doesn't work. So that was the study so I contacted the Lancet. The Lancet said, we're not interested because in the Lancet we usually don't publish letters like that. Okay. So we went to the Médecine et Maladies Infectieuses, which is the French Journal of Infectious Disease. And, no evidence of the activity of hydroxychloroquine. And I mentioned the reason why it didn't work. And, it was online March 2020. It was the most cited publication and the most downloaded publication this review ever had. And my colleague in the US, which were writing the guidelines in the US for COVID-19, told me, well, your data were important because this make us realize that hydroxychloroquine may not work. But still, a lot of people continue to use that drug, although I didn't want to be involved in any study with hydroxychloroquine. Well, we also published another study about another drug, remdesivir. I can spend a lot of time talking about that, because at that time also, France was the only country where remdesivir was supposed to be not working, and it was almost very difficult to use it, despite a randomized clinical trial done by the Insight team, which I mentioned earlier. I knew the team, I knew their work, and they said, no, they said, well, this drug provided benefit. But in France, people were reluctant about these drugs. I don't understand really why. There was many studies done by the ANRS at that time with mixed results. But then we, again, looked at what we've done in our unit, and we compared what we've done in our unit to what was done in other units where they didn't use remdesivir, and we found a very significant reduction of mortality. So, well, I'm going to skip that. I'm going to finish now. I know I'm late. Sorry for that. To explain why I'm now doing research on sexually transmitted infections. It was because during these studies in PrEP, we had a lot of people with a high number of sexually transmitted infection, and we said, well, we have to do something about that. And because these people were quite captive, because you see them every 3 months when they are on PrEP to detect HIV and STIs, and people all the time were not any longer concerned about HIV because they knew the PrEP was working very well. They were concerned about the other STIs. And so, well, in the wake of the end of the Hypergate trial, we said the participants were so happy about the results, they were ready to do anything that I would ask them to do. And so I told them, would you agree to be enrolled now in another study to test an intervention to prevent STIs? And again, looking at the literature on STIs, because not to mention that I didn't know anything about STIs before, I said, well, there is a drug that seems appropriate to prevent STI. It is doxycycline. And I selected doxycycline because of its activity against syphilis and chlamydia, and also because it has been used for the prevention of other infection like Lyme disease or leptospirosis. And I thought, well, at least we know something about how to use it for prophylaxis. These are a number of studies we've done during the PrEP studies to address the issue of STIs, not only bacterial STI, but also viral STIs, HPV, HHV8, or and others. But I'm going to skip that. And then we did a trial with post-exposure prophylaxis with doxycycline to prevent STIs in the Hypergate trial, which was published in 2018, a year after we got the results, because we were concerned about how people would use these results. So the trial was a simple design, doxycycline given after sexual intercourse, because people were already using PrEP before, so that was maybe too much, or no PrEP. And what we found was a reduction of the incidence of STI in blue with PrEP, but what was striking was the effectiveness on chlamydia with a reduction of nearly 70% and 80% with syphilis. No effectiveness on gonorrhea, probably because of the high rate of gonorrhea resistance, which we know was already there. So to confirm these results, we presented this year at CROI a larger study looking at doxycycline first, and also the 4C-MenB vaccine, because we knew the effectiveness on gonorrhea was not good enough, so we wanted to know whether a vaccine for meningococcal disease, which is very similar to gonorrhea, could cross-protect for gonorrhea. And so the results very quickly. Doxycycline, we confirm the effectiveness on chlamydia and syphilis because we combine the endpoint here, and you can see more than 80% reduction of chlamydia and syphilis when people use doxycycline post-exposure, so after sexual intercourse when they didn't use a condom. When you look separately at the two similar effectiveness, chlamydia or syphilis. Gonorrhea, there was a small decline in incidence, but not great, 30%, but still. But unfortunately, looking at the vaccine, despite retrospective studies showing that the vaccine could provide a 30% reduction in incidence of gonorrhea, in a randomized prospective study, there was no effect that we could detect on the primary endpoint and on the other secondary endpoints. So now we are trying to understand what was going on, and we are working with immunologists to try to assess immune responses and to see whether we can identify a subgroup of people who have an immune response to the vaccine, which may give protection for gonorrhea. And to do that, what's interesting is I'm going to work with the person in Italy who actually invented the Bexsero vaccine, and the group at GSK. The GSK company is very interested about the results, and they have all the assays set up in their lab to look at, very precise immunologic endpoints. So we are setting up a collaboration with them to look at that. So in conclusion, and sorry for being a bit long, but I would say for the future perspective, that are the advice I could give you. It's very important to maintain large collaboration. International collaboration are very key. Also working with networks is critical. Network within the NRS. We are lucky enough in France to have agencies funding research on infectious disease, and also working with the industry, because you learn a lot working with the industry, and you can have access early on to drugs that can be very useful. When we did our study with PrEP, with tenofovir FTCs, because all the work I've done before with Gilead that agreed to give me the drug for PrEP, because at that time, Gilead didn't care at all about using their drugs for PrEP. They wanted to use their drug for treatment. It's only because of the results of the studies that eventually now they are very interested to develop drugs for PrEP, but initially they didn't care about it. We had a fight actually with them about the patent for using TDF-FTC for PrEP. I can talk to you about that later if you want. So we are continuing research on opportunistic infection, on TB, and TB with the help of Olivier and his network, trying to assess and reduce the mortality of TB meningitis in many countries in Brazil and Africa. We are starting again with Olivier a large multinational, multi-country trial looking at doravirine versus dolutegravir to treat initial infection with HIV. I mentioned the study we are doing on PrEP with long-acting agents and also trying to do implementation research. That's really important now to make this drug available to those who need it. With STIs, I work with GSK now on a new vaccine for gonorrhea infection, which seems very promising. So let's wait for the result. This trial is fully enrolled now. We are, very eager to see the results. And I think the next really frontier in HIV research is the remission, the cure of HIV. And now, back again to my former colleague, Yves Lévy and Rodolphe, who is here, trying to see whether we can, design a study to try to induce HIV remission. So let's hope this is going to work. Develop new partnership is clearly very important. I mentioned the new partnership we're going to develop with GSK now on better understanding the immune response to the vaccine. And I think what's great also is to have in your group young, new and brilliant investigators who can help you to do all this work, because all this work is not my work. It's the work of a large number of people. I'm lucky enough to have a large group also in my department. And I mentioned not only nurses, but also not only doctors, but also nurses, research assistants. So the work that I presented here to you today is not my own work. It's the work of a very large team. And thank you for your attention. And I close here with the different partners we had over the years. And the Gates Foundation, for example, agreed to fund for the first time a study in Europe on PrEP. That was also the reason why we were able to conduct the study. So thanks very much for your attention.